coming back to our biology class. Today also let us start our class with few amazing facts of our body. The first amazing fact is, suppose if you take the example of any body part, it will work with the help of the oxygen. That means whenever the oxygen is supplied to that particular body part, then only it will work. So the oxygen is supplied to the different parts of the body with the help of the blood. But there is one body part which does not need the supply of blood. So which body part that is? Cornea. So where it is present? It is present in our eye. So here the cornea is the part which is present in our eye. It takes oxygen directly from the air. This is the one of the amazing fact in our body. Next, coming to the second amazing fact, the nose can able to identify the 50,000 sense. 50,000 sense. That is the one more amazing fact. Coming to the other fact, so you know children, for the process of digestion, our stomach produces the acid to digest the food materials. So that acid can able to dissolve the metals also. That acid can able to dissolve the metals also. Next, the last but not the least amazing fact is about the seahorse. Children, you heard the word seahorse, children? Yes, you heard the word seahorse. Usually, the reproduction will be carried by the female in each and every living organism. But here, the reproduction in seahorse, that means the male seahorse can give birth to the young ones. Usually, normally in almost all the organisms, the female can give birth to the young ones. But whereas in the seahorse, the male give birth to the young ones. So these are the amazing facts of today's session. Next, coming back to our class. So in the previous class, we discussed that who discovered the cell and who are the different scientists who contributed for the cell discovery. Is it the children? So who are the different scientists who kept their findings for the research of cell? Johnson, Robert Hooke, Robert Brown, Sheldon and Schwann. In the previous class, we also discussed about the unicellular organisms and multicellular organisms. Unicellular organism is the organism which consists of single cell. Multicellular organism, the organism which consists of more than one cell. So in unicellular organism, the single cell able to perform all the functions. But whereas in the multicellular organism, there are many cells which helps to perform all the functions. We also discussed so Robert Hooke written a book called Micrographia. Micrographia. And some other scientists who studied about the microorganisms, that also we discussed in the previous class. Who are they? Athanasius Kitcher was the scientist who introduced about the cell. That means he found out the plague. And Swammerdam was a scientist who found the red blood cell. He was the first person who found the red blood cell. And Antony von Leeuwenhoek who observed the different microorganisms under the microscope. Later, Robert Brown who introduced the cell. So, this we studied in the previous session. In today's session, we will learn about the procedure to observe the onion peel cells and human cheek cells. So, here, before going to learn the procedure to observe the onion peel cells and human cheek cells, we have to know the preparation of the temporary slide. So, how we have to prepare the temporary slide? Suppose if we have to observe any structure internally, means we have to get the thin section and we have to keep it on the slide, and we have to add a drop of water, we have to cover it with cover slip, and we have to see under the microscope. So this is the temporary slide preparation, but it's not the permanent. Later, to observe the onion peel cells and on human cheeks. So in the previous class, I told that Robert Hooke observed the thin section of the oak or cork. So it resembles the dead cells. Just like that, one more activity was given in our textbook about the thin section of the matchsticks. Do you know, children, matchsticks? I thought you all know. So here is the picture, see. So here also they gave the set. We have to cut the thin section of the matchstick. When we have to cut, we have to soak it for half an hour. After half an hour, we have to collect the matchstick and we have to take the thin section. We have to observe under the microscope. Then what will happen? So whenever we observe this thin section of matchsticks, whenever we observe the thin section of the cork also, here we observe the dead cells. Here we observe the dead cells. Usually we will find the dead cells in the sclerenchyma. Here usually we will observe the dead cells in the sclerenchyma. Next, we have to do the two activities today, that is to observe the onion peel cells and to observe the human cheek cells. So whenever we are observing the onion peel cells and human cheek cells, so onion peel cell is nothing but the onion is nothing but the plant. So here we are getting the one, here we are observing one plant cell and one is the animal cell. So now we will discuss, children, what is the procedure to observe under the microscope.
Onion and cheek cells. Onion peel. To do the experiment we require cover slip, glycerine, glass slides, onion, watch glasses, distilled water, saffron in solution, forceps, brush, needle, compound microscope, blotting paper. Procedure. Take some distilled water using a dropper and put it in a watch glass. Take half a piece of onion from the watch glass and remove a leaf from the onion piece. Remove a piece of transparent onion peel with the forceps. Put the epidermis into the watch glass containing water. Take some saffron in solution using a dropper. Put a few drops of saffron in solution into another watch glass. Using a brush, transfer the peel into a watch glass containing saffron in solution. Keep the epidermis for 30 seconds in saffron in solution so the peel can become stained. Using the brush, take the peel from the saffron in solution and put it into the watch glass containing distilled water. Using a dropper, take some glycerine and put 2 to 3 drops of glycerine in the middle of a dry glass slide. Using the brush, transfer the peel to the slide containing glycerine. Take a cover slip and place it gently on the peel with the help of a needle. Remove the extra glycerine using a blotting paper. View the slide under the compound microscope. Observations. A large number of regularly shaped cells are lying side by side. Each cell has a distinct cell wall. A distinct nucleus is present on the periphery of each cell. Lightly stained cytoplasm is observed in each cell. A large vacuole is present in the center of each cell, surrounded by the cytoplasm. Conclusion. All cell walls and large vacuoles are clearly observed in all cells, and the cells in the observations are plant cells. Human cheek cells. To do the experiment, we require toothpick, cover slip, methylene blue solution, glycerine, glass slide, distilled water, brush, needle, compound microscope, filter paper. Procedure. Take some distilled water using a dropper and put it in the middle of the glass slide. Take a clean toothpick and gently scrape the inner side of the cheek. Rub the scrap on the glass slide that contains water. Mix the mixture using a needle and spread it. Take methylene blue solution using a dropper and add a few drops to the mixture on the slide. After 2 to 3 minutes, remove excess water and stain using a blotting paper. Take glycerine using a dropper and add a few drops of glycerine to the test mixture. Take a clean cover slip and lower it carefully on the mixture by using a needle. Using a brush and needle, press the cover slip gently to spread the epithelial cells. Remove extra liquid around the cover slip using a blotting paper. View the slide under the compound microscope. Observations. A large number of flat and irregular shaped cells are observed. The cells do not have a cell wall. However, each cell has a thin cell membrane. A deeply stained nucleus is observed in the center of each cell. No prominent vacuole is observed in the cells. Conclusion. As the cells observed do not have a cell wall or a prominent vacuole, the cells of the specimen on the slide are animal cells.
So far we observed how we have to observe the onion peel cells with the experiment. How we have to observe the onion peel cells? The requirements need for to observe the onion peel cells is slide, onion and cover slip and microscope. So we have to cut the thin section of the onion peel and we have to keep it on the slide. We have to add a drop of water. We have to cover it with cover slip. Then we have to observe it under the microscope. Whenever we observe it under the microscope, we will observe the rectangular like structures which consists of the similar cells. So this is the procedure which we have to follow to observe the onion peel cells. Next, so here whenever we observe the onion peel cells, so it belongs to the plant cell. So we can conclude that so far we observed one plant cell. Next, coming to the next activity, so the procedure to observe the cheek cells. So usually here is the opportunity to get the cheek cells from ourselves. So here what we have to do is the requirements need for this experiment is slide, plastic or wooden spoon, cover slip, water, microscope. First of all, we have to wash our mouth 1 or 2 times. We have to rinse our mouth with water. Next, we have to take the wooden spoon or the plastic spoon. in order to get the saliva from our cheek. So we have to collect the amount of saliva from our cheek. So here we have to follow the precaution is we should not scrap our cheek again and again. If we scrap our cheek again and again, it may cause the wound for us. So gently we have to scrap our cheek. So we have to scrap our cheek and we have to collect the content with the spoon and we have to keep it on the slide. Whenever we keep it on the slide, afterwards we have to cover it with the cover slip. We have to cover it with the cover slip. Then we have to observe under the microscope. Whenever we observe under the microscope, here what will happen? So here also we find the rectangular shaped cells. Rectangular shaped cells. So here this is the example to observe an animal cell. So in this session what we observed? We observed the onion peel cells and human cheek cells. But whenever doing the human cheek cells, we have to follow the precaution because if we scrap our cheek again and again, it may cause wound to us. So far, we discussed the activities to observe the onion peel cells and human cheek cells. Did you find any difference, children, whenever we see the structure of the onion peel cells and the human cheek cells? Yes, we find one difference. So what is the difference? Suppose if whenever we find the onion peel cells, we'll observe that there are two layers surround. There are two layers. That means each and every cell is surrounded by two layers in onion peel cells. But whereas in human cheek cells, only the single layer is present. So here the cell membrane. So the outermost layer of the any cell is called as the cell membrane or plasma membrane. If we observe the onion peel cell, the outermost layer is called the cell wall. Whereas in the human cheek cells, the outermost layer is called as the plasma membrane. But the plasma membrane is present in both the plant cell and animal cell. But whereas the cell wall is present only in the plant cell, it is absent in the animal cell. That is the main difference for plant cell and animal cell. So here this plasma membrane gives size, shape to the animal cell. But whereas the cell wall gives the size, shape to the plant cell. So that is the main difference between the plant cell and animal cell. So what we discussed today? We discussed the procedure which helps to observe the matchstick. So what we discussed in that matchstick? We have to collect the matchsticks. We have to soak it for half an hour. Later, we have to cut the thin sections and we have to observe it under the microscope. Whenever we observe it under the microscope, we will observe the dead cells. We observe the dead cells. Next, coming to the procedure to observe the onion peel cells and human cheek cells. So, we have to remove the outermost layer of the onion and later we have to collect one part of the onion. We have to collect the thin section and we have to keep it on the slide. Whenever we are keeping it on the slide, we have to take the precaution that you should not fold. If it is folded, we are unable to see the structure of the onion peel cells. Then we have to observe under the microscope. Then we observe the onion peel cells. Like that only we observe the human cheek cells also. In human cheek cells, we have to scrap the material, not saliva, material from our cheek which helps us to observe the human cheek cell. So the outermost layer of the animal cell, that is the human cheek cell, is the plasma membrane, whereas the outermost layer of the plant cell is the plasma membrane. So whenever they are asked the questions, the question which they can ask regarding this topic is, what precautions we have to take whenever we are doing this human cheek cell experiment. So what are the precautions which we have to follow? We should not scrap the cheek hardly because it may causes the wound. Okay, the children. Next, coming to the animal cell, that plant cell, whenever we are observing the onion peel cell, so the thin section should not fold. So here the procedure is same to observe the onion peel cell and human cheek cell. But in one experiment we are keeping the onion peel, but whereas in another experiment we are keeping the scrapped material which we are scrapping from the cheek that we are observing. So this is the difference between the onion peel cell and human cheek cell. Whenever we are doing the experiment, we have to take the necessary precautions as already we studied in our first lesson. And we also know which membrane gives a structure to the animal cell, plasma membrane. But whereas for the plant cell, the cell wall gives the structure. So these are the activities which we discussed in this session. Let us recall what we discussed from the first class itself. We discussed that the different branches of biology. What is biology? Different branches of biology. Who is the father of botany? Who is the father of zoology? Who is the father of microbiology? And who discovered the cell? Who discovered the cell? Robert Hooke discovered the cell. Like that, 4 scientists were there who kept their experiments, who conducted their experiments on the cell. And we also discussed about the 5 more scientists who contributed to who discovered about the cell. Who are they? Jacob, who introduced the compound microscope, and one more person, Robert Hooke, who discovered the cell, Sheldon and Swan, who discovered the cell theory, Robert Brown, who discovered the nucleus. So here Sheldon and Swan introduced the cell theory also. What is the cell theory? Each and every living organism is made up of the cell. All the new cells are produced from the pre-existing cell. So Sheldon and Swan introduced the cell theory. Later it was modified by whom? Rudolf Virchow. So in the next session we will discuss about the nucleus. How we have to observe the nucleus in the onion peel cells and human cheek cell. So whatever experiments we learned so far, those are the temporary slides preparation, but not the permanent slide. So in the next session we will learn about the significant observation by Robert Brown and how to observe the nucleus in both the onion peel cell and human cheek cell.